Okay, today we're going to talk about kidneys and go over the anatomy of kidneys. We're not going to do much physiology today, that'll be saved for lecture. So first let's just start off with this model. I want to go over where the kidneys are located. So the kidneys are located in the posterior aspect of the visceral cavity, and they have their own cavity called the retroperitoneal cavity. The cavity in the front is called the peritoneal cavity, and that's where our GI tract is and accessory organs are, our liver, pancreas, so on and so forth. So we have to remove all of that to see our kidneys. Really, our kidneys could potentially be more accessible from the back because what's in the posterior aspect are the lower ribs and a lot of muscle tissue fascia. So our kidneys are located here. Your left kidney is a little bit higher, sits a little bit higher than your right side kidney, and that's because the liver takes up a lot of room on this side. Our spleen is above your left side kidney, and the adrenal glands are on top of the kidneys. Our adrenal glands is what provides hormones such as epinephrine and norepinephrine. All right, so that's the kidneys here, and then you can see the ureters coming out. So let's go to a zoomed in view and look at some more anatomical structures from a zoomed in view and take it to the next step. Okay, this is just a little bit more of a zoomed in view of the kidneys. Here we can see a good, decent outline of the renal cortex. We're going to go more in depth on the renal cortex and then the renal medulla right in here, and we're going to basically go more zoomed in, so that we can see some of the microscopic activity that's occurring in the kidneys. But this is just a good view to show how our abdominal aorta comes down and supplies the blood supply into the kidneys from our renal arteries, our main renal arteries, and then our main renal vein, bringing blood back from the kidneys up to the inferior vena cava. Here we can see our ureters coming down, and remember our ureters come from the renal pelvis, so the renal pelvis empties into the ureters, and that brings urine, waste products, and any other sort of substances that get secreted like ions, calcium, sodium, potassium, things like that. And H2O, of course, gets secreted here and then stored in the bladder. This is a good view of the bladder. In the PowerPoint, I go a little bit in depth on the tissue of the bladder, how it's a smooth muscle, it's very elastic in nature, and it has these structures inside similar to the inside of stomach called rugae. This bladder right here is specific as a female bladder. You can tell because it has the female portion of the sex organs here. But this is just a good example of what the inside of the bladder looks like. Okay, so now we're zooming in a little bit farther, or a little bit deeper, I should say. So our ureters come out right here. This is our renal pelvis. So the renal pelvis collects all of the urine that's coming out of the renal medullas, and our renal medullas are right through here. The renal medullas contain a large majority of the collecting ducts and some portions of the nephrons. So nephrons are microscopic structures that reside mostly, the majority of the nephron exists in the renal cortex, and this is really where filtration occurs. So filtration begins in the initial portion of the nephron, which is called Bozeman's capsule or the renal corpuscle, and this mostly exists here in the renal cortex, and we'll talk about those nephrons here in a little bit more detail. Also in the PowerPoint I go over those in more detail, but this gives you a nice little zoomed in view of what the renal medulla looks like and the renal cortex look like. And we have a couple million nephrons that exist within the kidney. So a lot of nephrons that exist, and a lot of filtration occurs that then they create our urine, and then empties from the collecting ducts into the renal pelvis and then down into the ureters. Now we got another zoomed in view. We're getting a little bit deeper here. This is one portion of a renal cortex and a renal medulla. So if we look at this, this is, for instance, this portion right here is right around this area right here, let's just say. So it's just one tiny sliver of a renal cortex and renal medulla. And it's showing two types of nephrons here, and we're going to go over these two types of nephrons, but there would be many more nephrons in this region. At this point right here, this is zoomed in at around 120 times normal size. So we're zoomed in about 120 times normal size to be able to see this nephron. And obviously, if I pull up this guy, so our kidneys are roughly about this size. And then, this portion right here would be real deep inside one of these renal medullas and renal cortex. So that kind of gives you an idea. So the renal cortex here, we have our nephrons. And if I look at one nephron, this nephron right here, this is called a cortical nephron. And you know it's a cortical nephron because what's called the loop of Henle, or the nephron loop, doesn't go very deep down into the renal medulla. So the initial portion here, we have the Bozeman's capsule, or the renal corpuscle, which this is a zoomed in view of the renal corpuscle, and we'll get to that in just a second. But this is where filtration begins. And the initial portion of filtration starts to flow into these tubules here called proximal convoluted tubules. And they're kind of called convoluted tubules because they kind of snake back and forth. Then it comes down to this loop of Henle, and this loop of Henle dips down into the renal medulla. And then the loop of Henle continues up into the proximal convoluted tubules. From the renal corpuscle through the proximal convoluted tubules, loop of Henle, and up to the distal convoluted tubules. I think I called those proximal. Sorry, this is distal convoluted tubules. This is where filtration occurs. So all of active and passive transport for ions such as calcium, sodium, potassium, hydrogen, H2O, all of this occurs in this portion. When the distal convoluted tubule empties into the collecting duct, there's really not much more diffusion that occurs. At this point, the urine just starts, it is what it is, it's at its substance. There may be some exchange of water, H2O, but for the most part, the urine just continues on down this collecting duct and eventually empties into the renal pelvis. All right, so here we get a zoomed in view. This is exactly where filtration begins. So the capillaries, so a lot of capillaries and veins come through here, and the capillaries come down into this region, into Bozeman's capsule, and these capillaries are known as fenestrated capillaries. So they're quite porous. They almost have like a Swiss cheese consistency, so that a lot of diffusion can occur, and then they can empty out into the proximal convoluted tubules, and filtration can continue to occur.
Okay, so functions of the kidney. One of the main functions of the kidney is filtration. So the liver does a good job of detoxifying our blood. The kidneys do a really good job of filtration and helping to maintain homeostasis of electrolytes. Another job is activating vitamin D and then gluconeogenesis. Remember, gluconeogenesis is utilizing protein and basically taking the protein molecule and altering the protein molecule so it can be a glucose-like substrate, so that it can be then used in glycolysis, and then eventually the product of the glycolysis can be used in oxidative phosphorylation. Remember that when gluconeogenesis occurs, that's because we don't have enough carbohydrates in our system. So when we're down on carbohydrates, gluconeogenesis has to occur, and we either use dietary protein or we harvest protein from muscle structures. So this function occurs in the kidney. The byproduct of this is a keto acid. This keto acid can be toxic to the body, so we really don't want keto acids to be too high. Okay, if we get keto acids too high, then we can go into what's called ketoacidosis, and then that can be some problems for our kidneys and our liver. Okay, so basic anatomy. I recommend you pause here and kind of take note of some of these structures. These structures are important. Make sure that you take note of the adrenal glands. This is where our adrenal hormones or adrenaline hormones are released, norepinephrine and epinephrine. Take note of the arteries that go, or arteries and veins that go to and from the kidneys. Our renal artery and renal vein, these are really important. There's a lot of blood supply that goes into the kidneys. Also take note of the ureters, okay? The ureters are what collect the fluid, eventually our urine, but collect urine and sends it down into our bladder for storage. Just a good cadaver view of the kidneys. And the kidneys are located in the retroperitoneal cavity. That's in the back of the human body, so behind our organs. So our organs are taken out here, our abdominal wall are taken out here, and you can see here that the kidneys are stored all the way in the back. So, and this is just another anatomical location. So underneath that 12th rib down to our accessory ribs. And then here's that cavity that I was talking about, the retroperitoneal cavity. Let me see if I can get a pointer tool out so we can look at some of this stuff. Okay, so here's our retroperitoneal cavity. This is the cavity, and it's behind the peritoneal cavity. The peritoneal cavity, remember, is where our organs are. This is our abdominal cavity. And then we have a vast layer of muscles that surround this cavity. Remember, the muscles in this area, in this region, are what provide the majority of stability. Okay, some important landmarks of our kidneys. So we have our renal cortex. The renal cortex is the outer portion of our kidneys. This is where the majority or the large portion of our nephrons exist, okay? And then the collecting ducts of our nephrons go down in through the renal medulla, okay? And then eventually it goes into the renal pelvis, and this renal pelvis is where a lot of the urine is collected. So filtration occurs through here, and then of the nephrons, there are loops that go down into the renal medulla. And we'll kind of do a zoomed in view here of some of that stuff as we go across. That's where filtration occurs. So all this blood supply here, all these capillaries, they surround the initial portion of our nephrons. And our initial portion of our nephrons are that Bozeman's capsule. And that's where filtration begins. And then those loops that go down of our nephrons go down into the renal medulla. And then eventually the collecting ducts go down here as well. So this is filtration. A lot of filtration occurs throughout this area. And the back and forth flow from the nephrons to our blood supply occur with ions. We're going to go over some of that stuff. But take note of all this. This is really important anatomy. You need to know this anatomy for your exam. All right, so this is basically just what nephrons are. So it's roughly about 1 million per kidney. Really important. Remember, a physician that is trained in kidneys and dealing with any sort of pathologies of the kidneys, known as a nephrologist. That's where we get the name from, is the nephrons itself. Two main portions of the nephron, the renal corpuscle and the renal tubule. We'll go over. So this is our renal tubule. Here, the renal tubule begins here with an area that's called the proximal convoluted tubule, and we'll go over that. And then there's a distal convoluted tubule, and then there's the loop of Henry, or the nephron loop. We'll go over that as well in detail. This is our glomulars, or Bozeman's capsule, renal capsule. Okay, so here's our example of the nephron. So this portion of the nephron is mainly in the renal cortex. And this portion of the nephron here is mainly in the renal medulla. And we have two different types of nephrons, and we're going to go over those two different types. But one type of nephron is called the cortical nephron. It's found mostly in the renal cortex. And then we have the juxtamedullary nephron, and I like to remember that name, juxtamedullary, by just saying just a medulla. I don't know, that's just my way of remembering it, but the majority of the juxtamedullary nephron exists down into the medulla. Not the majority, just the majority of the loop itself in the collecting duct. Okay, so these three major parts, I'll let you read this slide. You know, I'm not a fan of just reading off the slide, but you can see the three major parts of the renal tubule: proximal, distal, convoluted tubules, and then nephron loop. This is where movement occurs back and forth to maintain homeostasis through ion flow. Some toxins get dumped into these, but once it goes into the collecting duct, there is no more movement back and forth. Okay, so these you're going to need to know for your lecture, for the lecture portion, not necessarily lab portion, but these are the cells that exist within the different areas. Know these types of cells. And by the way, form follows function. Okay, so the form of these cells follows the function that is occurring in that area. All right, a lot of movement back and forth between these simple squamosal cells. These are simple squamosal cells, and these exist in the left nephron loop. So transfer of fluids and transfer of ions occur very readily in this area. Simple columnar, okay? Simple columnar and simple cuboidal cells exist in these areas in the proximal tubules, okay? And the distal tubules. And a little bit harder for things to basically transfer. Okay, so just know these, the nephron loop is a descending and an ascending loop. Our descending loop obviously is here as it goes down, and ascending loop as it goes up. These are important to know because different types of ions flow back and forth. And we're going to get to that. There's a slide with a good picture. And then collecting ducts, okay. Basically, this is where fluid begins to dump into, and these collecting ducts end up in the ureters eventually as they go through the renal medulla. Okay, really good illustration here. This is a cortical nephron, and this is a juxtamedullary nephron. We have more cortical nephrons than we do juxtamedullary nephrons. You can see how far down the juxtamedullary nephrons go. This is just a good slide to see how it sits. Okay, and this is, if we just take one little area here of our kidneys. These are the two classes of nephrons here. So go ahead and read this slide. You can pause. Feel free to pause as I go through this because I'm going through relatively fast. All right, another good example of the cortical nephron versus the juxtamedullary nephron. This has a lot of good anatomical information. So take note of this slide. Read over this slide. Okay. Okay, so capillary beds exist around our renal tubules, okay? And they are really important. This is where transfer of ions occur. This is where filtration occurs. The physiology of the kidneys. Roughly around 180 liters of fluid is processed daily, okay? That's a lot of fluid that's processed, all right? 180 liters. 1.5 of that urine is formed. All right. Vast majority of that urine ends up being H2O. All right, this is that slide I was telling you about. This is the physiology of a nephron, okay? This is actually what's occurring. This is what's transferring through. We have active transport going on. We have passive transport or passive diffusion, so active diffusion, passive diffusion going on. And then this is the regions of where this goes on, okay? When we get into the collecting duct, there's really not much ion flow that's occurring. We have some urea that transposes out or diffuses out, and then some water that diffuses out. But the majority of ion flow occurs here. Remember, this is all simple squamous, and then we have our simple cuboidal throughout this area here. All right, just a good zoomed in view. I'm going to let you guys take a moment here. Know this anatomical structures here. This is that Bozeman's capsule. This is where filtration begins. Remember, filtration begins here. And then the capillaries go and flow around the rest of the nephrons, around proximal convoluted tubules, the loop of Henry, or Henle, excuse me, and then the distal convoluted tubules. Really good view here of the capillaries that flow into Bozeman's capsule and how filtration begins. A lot of this stuff is going to be important for your lecture, okay? So not so important for our lab. These are cells here. This is the nucleus of the cell, and it surrounds the capillary. And then really just a good electron microscope view of it. I just thought this was pretty neat. Okay. But take note of the foot processes, the pedocyte cell body. This is where filtration begins here. Once again, this is mostly for your lecture version, not for lab. Quick, urea. If we have a lot of gluconeogenesis occurring, well, then we have a lot of urea that's produced. Okay. These are just different ions that are solutes in urine. Okay, obviously sodium and potassium play a major role in electrolyte. Same with calcium. These are very, very important ions for just normal occurrences in the human body. Sodium and potassium play a large role in electrical conductivity. Same with calcium, especially in the heart and the bundle of His. So remember our ureters, if you remember from that one slide, this is what comes out of our renal pelvis and then leads down to our bladder. And they empty out into our bladder, and our bladder is the main, sort of like a main holding tank for our urine, and helps to hold before we can basically empty out our bladder. And that's called micturition. Okay, so here we have our bladder. This is obviously male. Take note of the sphincters. We have an internal sphincter. We have an external sphincter. Okay, excuse me, external sphincter here, internal sphincter here. And then down here is our urethral orifice. This is our urethra. And then female. Urethra is much smaller, but still we have an external sphincter and an internal sphincter. So here's our external sphincter and then our internal sphincter. And then I'll let you read this slide. Once again, this is mostly for lecture, so slides like this. So just take note of these slides, take some notes on it. Okay, what is the capacity of our bladder storage? All right, how much can we hold? Roughly about a pint. Obviously, larger individuals can hold more than smaller individuals. And then this is just a good scan showing our urinary bladder, our renal pelvis, the ureters. These are renal medullaries are here. Okay. All right, and this talks about the sphincters that I just pointed out earlier. So, important note here for your lecture portion, the external sphincter is voluntary. The internal sphincter is involuntary. Okay. So this is controlled by our parasympathetic. This is mostly controlled by our sympathetic. Micturition. This is what is urination. This is what we call urination. This is the scientific name for urination, micturition. Okay? You need to know that for your exam.